Okay, so welcome to this next video in the playlist on cancer. In this video, what we're going to do is we're going to begin a bunch of videos which are going to be on the topic of apoptosis. Now, in this video, we're going to introduce the most basic enzyme which undertakes apoptosis, which is responsible for apoptosis, which is the caspases, which isn't just one enzyme, by the way, it's a whole family of enzymes. So we're going to look at the caspase enzymes. Okay, so I'll put that as the title, caspase enzymes. Right, now, firstly, what I want to discuss with you is firstly what the name means, where the name comes from. I then want to discuss with you what a caspase enzyme actually does, and then I want to discuss with you how you go from having a pro-caspase to having a caspase, and then some basic models of what's going to happen in apoptosis. Okay, before we actually discuss the apoptotic pathways, both intrinsic and extrinsic, in more detail. Okay, so firstly, what does caspase actually stand for? Well, this is what it stands for. The full name of a caspase enzyme is a cysteine dependent, aspartate dependent, aspartate directed protease. So aspartate directed protease. So cysteine dependent aspartate directed protease. That is what caspase stands for. So how on earth did they get caspase from that full name? Well, they took the C from cysteine. So that's where they got the C from. So C came from cysteine. Asp came from aspartate, here, asp for aspartate, and then the A's came from the protease back here. So that's where they got caspase from, cysteine dependent aspartate directed protease. And now that tells us what caspase enzymes are. And by the way, I should probably mention now that there are 12 different caspase enzymes. So it's not just one enzyme. It's a whole family of different enzymes. But they are all cysteine-dependent aspartate-directed protease. So they're all proteases. Now, what's this cysteine-dependent aspartate-directed? That's what I want to talk about now. So let's start off with this cysteine dependent. What does that mean? And then we'll talk about what aspartate directed means. Okay, so let's say this is a caspase enzyme here. Okay, and this is the active site of the caspase enzyme here. Basically, in the active site of the enzyme of a caspase, the active site of a caspase enzyme, you have a cysteine residue, and the cysteine residue is essential for the protease activity of this enzyme, which is why it's called cysteine dependent. So let me just remind you of the structure of a cysteine amino acid. So let's firstly just draw the basic structure of an amino acid. Here's the amino group, here's the alpha carbon, here's the carboxyl group coming down here. There we go. We've got a hydrogen coming off the alpha carbon, and then we've also got a methylene group with then a thiol group sticking on the end, which is a sulfur bonded to a hydrogen. This is the structure of a cysteine amino acid. So it's got this sulfur group sticking off the side. And basically, you have this sort of an amino acid residue in the active site. So let me circle this. Okay, so this entire thing is a cysteine amino acid here. Let me bring this out a bit. So this is a cysteine amino acid. And you have one of these in the center of the active site of these caspase enzymes. So that is where the cysteine-dependent portion comes from in the name. Now, let's turn our attention to aspartate-directed. What's that all about? Well, basically, these proteases don't just cleave N in any old place on a protein. They have a very specific recognition sequence in the protein which they will cut at, basically. Okay, so remember that proteins are polypeptides. So they are amino acid after amino acid after amino acid polymerized together. So basically, this protease is designed to recognize the sequence D, E, V, D, DEVD. And basically, this is a sequence of amino acids. So to understand what this means, you need to you need to know the single letter amino acid code. So you need to know that D stands for aspartate or aspartic acid. So aspartate. E stands for glutamate, so glutamate, and V stands for valine. So this is effectively aspartate, glutamate, valine, aspartate. Okay, so this is valine. Right. And the caspase enzyme is going to recognize this site, and basically it's going to cut here and here. So it's going to cut the bond between this aspartate here and whatever amino acid was here, and this aspartate here and the amino acid on this side. Okay, so let me draw out the full structure of this to give you a bit more intuition for what we're talking about here. So we'll start off with this aspartate here. So this aspartate has a peptide link with the next amino acid along. So there is its amino group that is bonded to the carboxyl group, or well, the carbonyl group now, of the next amino acid along, basically. So this is the start of our DVD sequence. So this is where this peptide link here is where the caspase enzyme is going to break. Okay, so here is our aspartate amino acid then. So here's our alpha carbon of the aspartate amino acid, and now let's put in our R group for aspartate. Now in the case of aspartate, it's just an ethanoic acid group here. So here is our aspartate R group. Okay, so it's a ethanoic acid group stuck off the side of the amino acid. Right, then you have the carboxyl group of this first aspartate, and there's that first aspartate complete. Now let's do the next amino acid along. It's glutamate next. So again we have our amino group of the glutamate amino acid, then the alpha carbon with a hydrogen off, and now we need to draw the R group for glutamate. Well, it's the same as aspartate, just with an extra methylene group in, so it's propanoic acid stuck off the side, rather than rather than ethanoic acid. Okay, and strictly speaking, since I'm calling them glutamate and aspartate, really they should be protonless. Glutamic acid is the name given to the amino acid when it's got the proton on this hydroxyl group. When it's donated its proton, because it's an acid and that's what acids do, the conjugate base, as it's called, the molecule without the proton, is then what glutamate is. Okay, so that really should be the oxygen without the proton, but never mind, everyone uses the two interchangeably. Okay, so here's our carbonyl group from the glutamate, which has this peptide link with our next amino acid on this sequence, which is valine. Now the structure of valine is that you have a carbon like so, and then you have two methyl groups coming off of this carbon. So we'll have them here and here, and then you have one hydrogen like so. So that's the structure of valine. That's the R group of valine anyway. And then here is the carbonyl group of valine. Then it's bonded in this amide link here with the final amino acid, which is again another aspartate. So again, you have this methylene and then the carboxyl group here. And this time I'll draw it with the O-, which is actually aspartate. So this is aspartic acid, that's aspartate. Okay, and then this carbonyl group is then linked in a peptide link on with the amino group of the next amino acid in this polypeptide. So this is where these caspase enzymes are going to cleave. They're going to cleave that peptide bond, and they're going to cleave that peptide bond. So they're going to come along, find these sequences, these D. Let me just label it up. This is D, E, V, D sequences. They're going to find these DVD sequences in proteins. They're going to bind to them, and then they're going to cut their here and here. So they're going to break proteins apart at these DVD sequences. So that's why they are called a protease. So that explains now their full name. Cysteine dependent, because the protease activity is dependent on this cysteine in the active site. Aspartate directed, because both of the cleavings occur next to aspartates. So you cleave the peptide bond next to an aspartate here, and you cleave the peptide bond in which this other aspartate is involved in over here. So that's why it's aspartate directed, and then protease, because you are breaking apart proteins. Okay, so that's all for this video. In the next video we'll discuss how you go from procaspases to caspases.